Okay, I'm going to do another recording here on signal transduction for hydrophilic hormones. And I'm going to selectively do hormones that are utilizing G protein coupled receptors that are tied to either the adenylate cyclase enzyme or the guanylate cyclase enzyme. And again, as in class, I said those two are very similar. So we're really going to have to do one of them and then show you the slight differences. So I'm going to start with a cell. I know it would be a little bit better if I had a little bit more room here, bigger board. My desk is in the way. I can't get out of the way much more. I'm going to put our receptor up here. Right? Remember, this is for hydrophilic hormones, and you know that because the binding site for the hormone is toward the extracellular space. Lipophilic hormones do not require that because they are lipophilic and can pass through the lipid bilayer to enter the cell where they will bind their receptors. And we'll do that maybe in a separate video. So I've got my triangle-shaped hormone molecule here, which has diffused out of the capillaries and into the interstitial space. Remember, this is the ISF out here, right, outside of our cells. It's just diffusing around, and by chance it comes in contact with our receptor molecule. Put an R on that for receptor, and binds, and we now have what's called the hormone receptor complex, which is going to initiate this sort of cascade of events, intracellular events. And remember, the goal here is to alter the types, amounts, or activities of cellular proteins, because that will change the function of this cell, which is what hormones do, right? Pick a hormone, pick a target cell. In some fashion or another, they're altering the types, amounts, or activities of cellular proteins to change the function of that cell. So and let me back up just a second. Remember, we do have a smaller number of receptors that are either channels, right? In other words, I would make this, I don't want to draw it on here and confuse the picture, but this receptor could literally be a protein that forms a channel as well. So it both binds the hormone and it's a channel. So that's the receptor channels. Or it could be an enzyme itself. They call those receptor enzymes. They're usually kinases. And we'll use that terminology again a little bit later in this scenario, so you'll know what a kinase is. All right? So this is the third type, and probably the most prevalent one, which is the G protein coupled. We call them recruiter receptors in class, right? And what they do is they recruit the other proteins to initiate the cellular response. So the first protein that gets recruited once we have the hormone receptor complex is the G protein. Right, so that's the G protein. Remember, it can be stimulatory or inhibitory, I or S. I'm just putting plus or minus there. I guess I could put I or S there, or S or I guess, right? But stimulatory or inhibitory, and it's stimulatory or inhibitory to the enzyme with which it associates, right? That also gets recruited. So let's put the enzyme here, and as I said before, it's either the adenylate cyclase or guanylate cyclase scenario that we're running through here, so I have to pick one. We're going to go with the one I did in class, which is adenylate cyclase. All right? Adenylate cyclase is the enzyme that cuts ATP to form the second messenger. All right? And so I can have my ATP substrate coming in contact with this enzyme to produce the cyclic AMP molecule. Okay? That's, in this particular scenario, that's our second messenger. Cyclic AMP acts on cyclic AMP dependent kinases. In other words, those kinases that require cyclic AMP to become active. And so we can show this in a couple different ways. Have it like this. So inactive kinases become, sort of arrows, active kinases, right? So the cyclic AMP is binding to, interacting with the inactive kinases to make them active kinases, okay? Now a kinase as we said before, is an enzyme that adds a phosphate group to particular amino acids. Of course, amino acids within the structure of a protein. Again, remember, we're trying to alter the types, amounts, or activities of cellular proteins. One of the ways to do that is to modify the protein structure. Remember, structure and function in a protein go hand in hand. So if we modify the structure by adding a phosphate group, we modify its function. So we have we have an active kinase, and we have, let's do this. Inactive proteins, right? And again, there are scenarios where phosphorylation does inactivate a protein instead of activate it, but that's sort of the exception. Usually we're taking a kinase to phosphorylate those proteins that are inactive to make them active proteins, right? And we'll stick a phosphate group on them here. It's just P with a circle on it to indicate that they've been phosphorylated. Okay? Now, we've essentially changed the function of the cell. So what are these proteins? So and I don't have room to sort of squeeze it all in here, but those proteins can be enzymes, channels, pumps, transcription factors, right? And again, so the first of that group, the enzymes, channels, pumps, would be altering the function of cells that are already in, the function of proteins that are already in existence, that have already been made. Altering transcription factors, yes, we're altering the activity of that particular protein, but what do transcription factors do? Well, transcription factors act on DNA to initiate transcription and translation. So again, with our hydrophilic hormone, we can alter the activities of existing proteins, channels, pumps, enzymes, but we can also initiate through transcription factors the production of new or different proteins from DNA through transcription translation, right? So again, our active proteins here, which ones become active will depend upon what this hormone was, what the kinases were that became active, and this will be cell specific, right? Certain cells express certain kinases, and those kinases will activate specific proteins within that cell. And so the response can be different in different locations, right? Because of differences in this whole signal transduction pathway. Okay? Now, so now we have usually an increase in activity of some sort, an added function for this cell, right? We went from inactive to active. Hormones generate those responses, but we don't want them to persist forever necessarily, right? We may want them to persist for minutes or hours or days, but and maybe we want them to go up and down in terms of the degree of activity. So how would we adjust this, right? Or what I try to do is I just go to the extreme. How would we shut it off? Well, you have to do a bunch of things to shut it off, right? So the first thing you'd want to do is actually get rid of the hormone, right? You'd have to degrade the hormone. And that can happen in a couple ways, but we can just sort of summarize and say it needs to be enzymatically degraded, right? We get rid of the hormone molecule, then the receptor would no longer be bound to it, and we would effectively stop activating this enzyme, right? So we'll just write degrading enzymes, right? They would tend to turn off this response. Okay, so we take the hormone away, right, through degrading enzymes that chew it up. But have we gotten rid of our cyclic AMP? No, right? Whatever we produced, and remember, enzymes function very quickly. We had amplification because one hormone binding might produce thousands and thousands of molecules of cyclic AMP, right? A bigger signal inside the cell, amplification. But we have to get rid of the cyclic AMP if we want to shut this off. Well, that happens through, and I'm going to use an abbreviation here, PDE. So PDE is an enzyme called phosphodiesterase. Phosphodiesterase cuts the ring structure within cyclic AMP that's irrelevant. But it enzymatically modifies the cyclic AMP, makes it into an inactive molecule, which does not act as a second messenger, and that's called 5' AMP. Right? That's inactive. Put that under here. Okay, so we've gotten rid of our second messenger. Great. So now the cell's going to stop functioning, right? Or stop doing what we stimulated it to do, right? Wrong. Because the kinases that were activated by cyclic AMP, right, have already phosphorylated inactive proteins to become active proteins. In other words, we haven't undone this phosphorylation. So to make our proteins inactive again, usually inactive, but the phosphorylation tended to be activating, but to make them inactive again, we have to dephosphorylate, remove the phosphate group. To do that, we need an enzyme. It doesn't come from outside the cell, it's inside the cell, and it's squeezed up here in the corner. It's called a phosphatase. So you want to think of a phosphatase as pretty much the exact opposite of a kinase. Kinases phosphorylate proteins, inactive proteins, right? Become phosphorylated, become active proteins. Phosphatases cleave this phosphate group off. Right? I'll put a negative sign here to indicate that it's going to remove that phosphate group to return these active proteins to the inactive state. Now we've shut off our response, right? Because we've deactivated our enzyme by removing the hormone here. We've gotten rid of our second messenger by making it an inactive byproduct of the cleavage of this molecule by this enzyme, phosphodiesterase. And we've undone the phosphorylation that occurred with our kinases that got activated through the phosphatase. Okay? So now we've sort of like shown how we can undo the activation that this hormone initiated. Okay? Now, if we were to go to a different cell, it might not be, or even a different receptor, it might not be adenylate cyclase that the G protein is linked to, it might be guanylate cyclase. Well, guanylate cyclase doesn't act on ATP, it acts on GTP. And then you don't get a product called cyclic AMP, you get a product called cyclic GMP. That's the only difference, right? And of course the product that produced here would be 5' GMP, right? But okay, so just substitute G's for A's, and you have a different scenario that could be linked to different kinases, through different hormones, whatever. It gives us some variety for how this cell would respond to different hormones, okay? Last thing I'll throw out here is that phosphodiesterase actually has exogenous, meaning from the outside, inhibitors. So those would be, this is PDEIs, right, from phosphodiesterase inhibitors. Well, a lot of us actually consume phosphodiesterase inhibitors on the regular. Things like, it's a group called methylxanthines, not so important, but it includes things like caffeine, theophylline, which is sort of the caffeine, quote-unquote caffeine in tea, and theobromide, which is a very similar molecule which is found in chocolate. So we consume these phosphodiesterase inhibitors, which block this enzyme, which means we would get less degradation, which means our cyclic AMP or cyclic GMP would persist, right, or persist for longer and actually maybe increase more than it would, which of course would tend to activate this pathway. It's sort of enhancing the response to this particular hormone, right? Because remember, this is inhibitory, the phosphodiesterase, right? Because it removes the second messenger. Well, if you inhibit an inhibitor, you activate the pathway. Sort of like, think about driving your car. If you inhibit your brakes, right, which inhibit the car, well, you don't want your brakes to not work because it means you're going to go faster, right? So that inhibitor would make this process be more robust and end up with more active proteins and a bigger hormonal response. Okay? I'll probably come back and do another one of these, but looking at swapping out the adenylate guanylate cyclase for phospholipase C, the other big alternative sort of enzyme that can be recruited, and it leads to a completely different intracellular pathway here.